Okay, so just for finish up the afternoon, I'm listed as talking about research and future perspectives. And I think this is just really what I wanted to do was to highlight, following a research discussion day yesterday, where we very much had a group of 50 individuals from around the world who not only specialise in research in hypothalamic hamartoma, but also related fields, to try and get some crosstalk, some cross-perspective, and also, and particularly in the afternoon, had quite an interesting, heated discussion about the merits of the various different techniques for intervention. So all I'm going to do over the next few minutes, and it really is a few minutes, is to summarise where I think we got to. So we started the morning off by discussion of etiology, and certainly that's something that Jack touched on this morning. There's been a lot of talk about genetics, perhaps a genetic etiology. We know that in Pallister Hall, which is about 10% of those, the HH population, if not less, there is a mutation known in GLE3, but what's happening with the rest of them? And if we had a sort of a handle, as we've seen in other epilepsies, about perhaps the genetic mutation, perhaps we could think about more targeted treatments. The most recent paper by Hildebrand and colleagues, as again Jack alluded to this morning, demonstrated somatic mosaicism, somatic mutations within the actual lesion itself. And it was 37% of the ones they looked at, which involved GLE3 or other components of the sonic hedgehog pathway, but it is not one, and there was more than one mutation in many individuals. So there doesn't appear to be a really targeted genetic cause at the present time for this. And therefore, then there's all this discussion about the role of the sonic hedgehog pathway. Is it one area, one part of it? Is it another part of it? Should it be inhibition or excitation? Certainly, there was a lot of discussion about that. So there was then discussion about how we could move forward, because tissue is becoming scarce, not least because of all the non-invasive techniques that are now being put forward to ablate or modify what's happening from the hamartoma. So there was talk about perhaps using DNA from CSF, also further tissue studies. How can we get tissue? Can we pool tissue? Can we cause a biobank, utilize a biobank? And then there was some discussion about whether we could learn from research in other, and I've put in quotation marks, brain tumours, because we know this isn't a tumour, it's a hamartoma. And part of the problem is this is not dividing tissue, and therefore trying to model it in vitro is quite difficult. And further, the issues about an animal model we also discussed, but again, without one specific genetic cause, it's difficult to know which way you'll go, let alone how you can utilise the tissue to generate any sort of model. There's the issue about what has been done on tissue, not least to look about why these lesions are so epileptogenic. And we heard earlier again from Jack, but also there was a lot of discussion yesterday about the work that's done with the cells, looking at the types of cell within the hamartoma and the relative excitation or inhibition that may be may be generated by those particular cells. And there's a lot of extending beyond that and looking at electrophysiological studies, not least from recording within hamartomas as well as outside, that gelastic seizures no doubt come from within the hypothalamic hamartoma, but it's when the other seizures arise, are they arising from within the hamartoma, or is it secondary epileptogenesis, about which in other types of epilepsy there's long been much discussion as to whether it exists, but within this particular syndrome it appears that seizures ultimately, if they progress, do arise from other areas of the brain, and this is acquired. It is not what happens at the outset of the disease. Certainly with much of the SLICE work, there's also looking at the maturity of the transport systems within each of the cells, the excitation and the inhibition, the maturity of the GABA system, and looking where else, some of the other medications that have been suggested, as was suggested by Jack earlier, that could be utilised, is looking at those pathways and seeing whether enhancement or indeed inhibition of those pathways could be looked at utilising medications that are already licensed for other purposes, so repurposing medication. For example, bumetanide. There is an imbalance of KCC2 and NKCC1. NKCC1, I understand, is relatively okay, but KCC2 is reduced, and that may have, utilisation of bumetanide may have an indirect effect on that. But it's all speculation at the present time, and we need to look systematically at what we find with in vitro studies to apply them to what we may do in vivo, not least looking at small cohorts of patients with consistent protocols, repurposing medication. We also heard yesterday, and we've heard today, about the imaging studies, and this I've taken today from Verina's work with regard to connectivity studies. And this utilised perhaps in the first instance to look at what effects may be occurring on the brain as a result of the seizures that occur with hypothalamic hamartoma, or even as part of the network itself, but also subsequently possibly in utilisation in possible surgical planning. But certainly there was a lot of discussion as to how this may be taken forward. We learned also, as I highlighted earlier, that there is very little known. We know about the high rates of cognitive and behaviour impairment, particularly in these children with ongoing seizures. But the issue is what the range of those abnormalities are, the actual specific phenotypes, and is there a specific phenotype to hypothalamic hamartoma. We've heard specifically about rage attacks. We found in our cohort 46%. Certainly when you're looking at studies in the literature, it doesn't seem to fit into a specific DSM-4 criteria, and therefore it may be underreported. But that aside, what we do know is that we need to look at these phenotypes in detail and then also look at possible interventions. Do standard interventions apply, or do we need to look more specifically at that? And of course, there's the whole issue about who is at risk of these cognitive and behaviour problems. Do they require specific intervention? But also, how do we prevent them, perhaps by determining those children early in their natural history that are at risk? And then I'm not going to emphasise again the wide range of interventions that we now have from a surgical point of view. And we heard very fast yesterday about all these techniques, and we've heard about some of them today. And one particular area that we haven't heard about today is focused ultrasound, which has not been applied in patients yet. This appears to be another ablative technique, but it is non-invasive. And there is a study being initiated in the United States for adults, for those 18 and over. But it is utilising focused ultrasound and MRI with targeted ultrasound to aim to ablate the lesion or generate heat in the lesion in a non-invasive way. So looks like the next stage on from the stereotactic laser thermal ablation. What was quite clear was, and what's been really interesting to me, and I'd be interested to hear from others, is having everybody in the same room who actually perform the different techniques. You know, there was a realization, as Dan and Jean have pointed out, that actually one technique does not fit all, and it's a case of deciding what technique may be optimal in certain circumstances. And therefore, yes, in certain centres there will be most experience with one particular technique, but in those centres that have a variety of techniques, they're becoming more experienced at utilising those in different circumstances. And of course, was highlighted earlier, we've all quoted the de Lalonde classification, but there are at least 2 other classification systems that people utilise. If we're all going to speak the same language, we need to decide what we're looking at. But what's specifically useful for this is then we can look at different types of lesion and decide perhaps what technique might most appropriate. And this is the algorithm that Jack had devised, but actually it's, we had a long discussion as to whether this was appropriate. It seems, there's lots of what ifs and if when, and, again, the more experience we gain with these techniques, the more experience we gain about what lesion may require what type of technique in which circumstance in each individual child. So where did we leave it at the end of the day? Well, I actually think we did come out with a a conclusion, we had a long discussion about how we may gain more tissue and how we may make best use of that tissue, and that can be quite a difficult decision. But there was talk about working towards perhaps a consortium to look at a biobank, a tissue biobank, and therefore a constructive way at looking at how we can utilise that tissue and put it to the best use. We also had a long discussion at the end of the day about how little we know about the pros and cons of all the techniques and how perhaps we should be consistent in data collection, but not least also try and make it as user-friendly in doing that. If we thought about a international observational study, so to speak, where we could collect information online, maybe some input from physicians, some input from parents and individual patients about seizures, cognition and behaviour, looking at online tools, then maybe we could all consistently collect that data and collect a cohort of information or information about a cohort with a variety of different techniques. That's not going to tell us whether one technique is better than another, but what it'll do is provide some baseline information as to which types of individual are being having intervention, and also whether there's any sort of evidence that the situations are improved or indeed remain the same. Perhaps even planning towards further studies in the future. And then the final thing that we came out with, and I have to say that this hasn't really been discussed, but actually in a wider scale, but I think needs to be looked at in the context, for instance, in John's the supplement that you've produced, which is about to come out, I understand, as to whether there does need to be any short discussion paper, the fact that where we stand at the moment. Now, it may be that that's in your supplement and I can't remember, so I'm afraid I'm standing here having not had that discussion with you. But, you know, as a result of yesterday and where we were all coming from, you know, it's something we need to think about and as maybe even a starting point for where we go in the future.